My name is Badri Adhikari. I'm an assistant professor at the University of Missouri, St. Louis, and today I'm going to talk about PDNET. The title of my talk is Deep Learning Protein Contacts and Real Value Distances using PDNET. PDNET, or Protein Distance Net, is a framework for deep learning protein interresidue distances. The framework consists of two things. First, it consists of a dataset of around 3.5 thousand protein chains as the development set. And second, it consists of all the deep learning code that you need for training, validation, and testing, all for predicting interresidue distances. The problem of protein interresidue distance prediction is similar to a problem known as image depth prediction in computer vision. In computer vision, for image depth prediction, the input is an image, typically a color image like this, from which we extract channels, typically red, green, and blue channels, RGB, which forms an input volume. It's a volume because you have each matrix, one matrix represents each channel. We have in total 3 channels. So the whole image now becomes an input volume. And the task in image depth prediction is we build convolutional neural network models which accept this input volume as the input and then predict as an output a matrix. Now this matrix is a predicted depth. Each pixel in this matrix represents how far or how close each pixel is to the camera. For example, in this picture, the lady's chin is very close to the camera, and we built a model that can predict very high values for around the chin region. The distance prediction problem is similar to this from the perspective of deep learning and machine learning. In case of proteins, what we have is a sequence as input from which we predict many input features. So for this project, it's not really important what features we predict because we use some commonly used standard features. But then what comes out of all of these predictions is the similar thing, a volume where we have the x and y axis representing the protein chain. Each dot represents pairwise features, and then we have n channels. Instead of 3 channels in images, we have n channels, and this n could be 3, 4, sometimes even up to 1,000 channels. Now the rest is similar. We are supposed to build a convolutional neural network model or any other deep learning model that takes as input this input volume and then produces an output matrix. Now instead of a depth matrix, now what we have is a predicted distance map as the output, where each pixel represents the actual distance between every pair of amino acids in the sequence. Say for example, we have a protein structure that looks like this. And let's say here's a distance map that corresponds to this protein structure. Predicting real value distances is very difficult. So for a very long time, we predicted contacts, which is simply distances less than 8 angstrom represented by a 1 and all others represented by a 0. So this is a binary version of the distance prediction problem, and for many years we've been doing this. Very recently, many groups have started predicting something known as distance ranges or distance bins. That is, what is the probability that a distance might fall within a range of 0 to 4 angstrom, 4 to 6 angstrom, 6 to 8 angstrom, and so on. We would like to call this as paradigm 1 and paradigm 2. In this work, we introduce a third paradigm, which is the paradigm of predicting real value distances. That is, we do not want to formulate the problem of predicting distances either as a binary problem or a multi-class classification problem, but rather as a raw real value distance prediction problem as it is in nature. So why do we need a framework for distance prediction? Currently, we rely on something known as co-evolution signals calculated from multiple sequence alignments to predict protein structures. But a protein sequence folding in a cell does not have access to co-evolution information. Andrew Sr. at DeepMind talks about this, and recently Professor Baldi's group also discussed this. So we may be wrong. It is possible that multiple sequence alignments are not the solution to the protein folding problem. We all know that we are very far from solving the protein folding problem. So having a framework enables us to investigate all the potentials and limitations at this merging superhighway of protein structure prediction and deep learning. Now the question naturally is, should we predict contacts, distances, or distance bins? In the PDNET framework, we discuss all three. We develop a method for contact prediction, a method for bin distance range prediction, and a method for real value distance prediction, and compare them. The focus of this work is real value distances and not contacts or distance range prediction. Now, in the protein structure prediction domain, it is more meaningful to predict inter-residue interactions than non-interactions. That is, we are interested in predicting all the shorter distances than longer distances. Now, this makes sense because from the perspective of a structure prediction and binding site prediction, it's more meaningful to predict interactions than non-interactions. Also, in 2014, Polastri et al. Showed that when we reconstruct models using predicted distances, we get more accurate models than using contacts or distance ranges. So we first attempted to predict real value distances using deep learning models, and it turns out that many of the distances, which are very larger in values, overwhelm a distance map which has very few short distances, which actually form the distance patterns in a distance map. Now, any typical loss function will first try to optimize these large distance values. So in this work, we propose the idea of reciprocating a distance map so that a true distance map that looks like this, when reversed, looks like something exactly opposite. So now in this distance map, smaller values correspond to the actual large distances, which are not very important, and the larger values, these larger values, correspond to the actual short physical distances, which are very important. So in this way, we formulate the problem as predicting real valued interactions rather than non-interactions. To illustrate that real valued distance prediction is promising, as a random example, we picked a protein from our test set and reconstructed 3D models using predicted contacts and predicted distances. And we found out that using real value distances can reconstruct much more accurate models than just using predicted contacts. Here, green is the true structure and orange is the predicted reconstructed structure. The PDNET framework is currently publicly available at this GitHub repository. With PDNET, deep learning models can be built in publicly accessible platforms such as Google Colab. The entire dataset is around 1 gigabyte and can be downloaded in less than a minute. All the input channels can be visualized in Google Colab, including the comparison of predictions with the true structures. Training a reasonably good model in a platform like Google Colab with a good GPU takes a few hours. I believe that the framework we have created, PDNET, will be particularly helpful for researchers with some machine learning background or even very rich machine learning background, but those who are interested in difficult problems like protein structure prediction, because many problems in deep learning today have very high accuracies, whereas in problems like protein distance prediction, the accuracies are not that high. And I also believe that in future, to extend this work, significant contributions can be made by focusing on three things. One is novel feature engineering techniques. Second is coming up with novel loss functions. And the third is coming up with specific deep learning architectures that may be particularly suitable for this specific problem of distance prediction. I would like to thank my graduate and undergraduate students at UMSL, the IT support team at our university, and Google, NVIDIA, the system, and the National Science Foundation. Thank you.